I'm going to talk today about our novel approach to DMD centered on the Notch pathway to modulate muscle stem cells. Start my video. Can you see this?
Yes, we can. Thank you so much.
Okay. All right. Why will I not go forward?
You just click on the screen and then try it again.
So this is a collaborative effort between my lab with Felipe de Suzalite and Mayana Zatz's lab and Joyce Epicito. And this comes about from two different directions. First, are studies with Mayana Zatz on a golden retriever some of the dogs in her colony escaped dystrophin deficiency and lived a normal lifespan. Those dogs increased the level of JAG1 in their muscle. And we were able to show that in zebrafish, and now I'll show you tonight, in mice, you can improve outcome by increasing JAG1 expression. In addition, the Zatz laboratory also came across and identified two patients unrelated to one another who were boys with Duchenne, no dystrophin, yet lived into their late 20s, into their 30s, were ambulatory for much longer. We did sequencing and genomics on those two different patients and came up with sequence variants in the Notch3 gene. So to test this, we decided to set up mouse models of each of these two different systems. We made an MDX mouse carrying one of the Notch3 variants, and we made a MDX mouse overexpressing human JAG1 in their striated muscles. The JAG1 expressing mice had stronger muscles, as shown by this slide here. The blue lines show that they have increased strength. The muscles were also larger. We did the same thing, and it's in the progress right now, on the Notch3 mice. We've done histology, open field test, fatigue tests, rotarod, startle response, and we're in the process of doing ex vivo physiology and histology and a few more things. But our original indications are that these mice as well have improved motor function and muscle size. What are we doing? What else have we done? We've done molecular consequences. That is, what is the interaction between Notch3 and JAG1 in vitro, I mean. We've derived IPS lines from each of these different patients. We've set up isogenic lines, and we're in the process of sorting these. Our progress is to publish the JAG1 paper, which is actually a publication almost ready to be submitted. We're finishing our Notch3 data collection. We plan to publish that. We want to translate these findings, but we're also in the process of discussions with biopharma to try to translate these findings into places where we can move this forward into patients. This could not have been done without the support of Defeat Duchenne, as well as other institutes, including the National Institutes of Health. And I would be remiss to not acknowledge the other people who contributed to this study who are listed in this slide. And I hope I did my little spiel in the 3 or 4 minutes allotted. Thank you.